Hey guys, hopefully you have watched the Edpuzzle I assigned yesterday that talks about the Miller-Urey experiment. If you haven't, I would really encourage you to go back and do that before you watch this, but either way, here we go. Okay, so as late as the 1800s, so we're talking like 200 years ago or less. Okay, so this is for the existence of humanity a very, very short while ago. A lot of people adhere to this idea called spontaneous generation. Okay, if something is spontaneous, it's kind of like out of nowhere. Okay, so like if somebody's really spontaneous, if somebody's described as a spontaneous person, that means they might do something unexpected, right? So spontaneous is kind of out of nowhere, and generation in this case is talking about the generation of life. Okay, so the making. Okay, so it's like out of nowhere, we're making new stuff. So this is the idea that life, living things, can be generated easily from non-living matter. Okay, and that might seem crazy to you because, believe it or not, you know quite a bit about science. Okay, so some of the classic evidence that they used for this idea was that meat and dead animals, which are basically meat, okay, if they were there for very long, just kind of out in the world, maggots would appear. And they're like, well, there were no maggots there before, and now there are maggots. I didn't see the maggots crawl over to the animal. I didn't see the maggots crawl onto the meat, but all of a sudden, no maggots, poof, maggots, right? Another example was that fleas appear when things are left with dirt. Alright, so these are kind of examples of them seeing those things. Another one would be like, you know, if you have a big storage grain pile, all of a sudden there'll be mice and rodents and things eating on them. And they came out of nowhere. I didn't see them there before, and here they are. Right? So some scientists started thinking about this idea, and they started to think maybe it wasn't correct. Okay, so we have Francisco Redi. I've heard Reedy. We're going to say Redi. Designed one experiment that he thought was going to kind of disprove this idea. Okay, so he designed an experiment to show that maggots came from flies rather than the decaying meat. Okay, so let's look at his experiment. So he has a sealed up flask. And I wish this didn't look like a maggot, but that's like supposed to be, I think, the piece of bone right there. Okay, so he's got a piece of meat in a sealed up flask with like a solid lid on it. He's got another one that has gauze on it, which is kind of like, it's like fabric. Okay, and one that's just kind of open. Okay, so you see no maggots grew on either of these, and maggots started living on this one. And he also saw that there was some things happening up here. There were some maggots that were growing up on top of the gauze, but not on the meat. Okay. But he also saw, hey, there were flies kind of hanging around. All right, so this is one of those things that worked a lot better when we were in class because I wanted to go over the scientific method with you again. But he showed that when the flies couldn't get to the meat, there were no maggots. Okay, so I'm just going to kind of try to talk through this like I would if we were in class, and hopefully it makes sense to you. If it doesn't, please reach out to me. I'd love, love, love for you to have a good grasp of these variables before you leave my class. Okay, so what was this independent variable? Okay, so remember the independent variable is the one that the scientist changes, the one that it makes different between the two groups. So we look back up at his experiment. What did he make different between these three kind of scenarios here, right? He changed the container's covering. Okay, it was either covered with a solid lid, a mesh thing, or no lid at all. So he changed the covering. Okay, and what depended on that? What was he looking for or measuring? Okay, so he was looking for or measuring the presence or absence of what? Okay, maggots. And what were 3 controlled variables, right? So what are 3 things that he kept the same? Okay, so he had the same meat, same kind of meat, the same size container. And we can't see it in this picture, but I'm going to tell you he would have had to kept time the same for all 3 too. Right? He couldn't have just sealed this one up and looked at it for 5 minutes and been like, yep, no maggots, but left this one out for 2 weeks, right? He had to have them all out for the same amount of time in the same conditions, right? So they also, we also could have said they should have had the same temperature, the same location, like don't want to put this one inside and this one out in the barn, right? That would be unfair also. Okay. Next page. Okay, then back up. Okay, people were not quite convinced yet. Okay, so they had this experiment and scientists were still kind of on board with spontaneous generation. Okay, another scientist. This one, you might not think you've heard of him, but you've heard of him. Okay, Louis Pasteur. Okay, so he showed that spontaneous generation was false. He had a little bit better experiment, and at the same time invented a very, very important process for humanity called pasteurization. Pasteurization, there you go. Okay, so it was known at the time that if you have a broth or liquid that contains any kind of nutrients. Okay, so maybe it's, broth for when you cooked a chicken and you kept all the grease or something like that. It's got nutrients in it. It's got good stuff. Okay, if you left it out, it would spoil and become full of microorganisms. Okay, so it would get brown and cloudy and icky because stuff would start to be growing in it. Okay, so let's look at his experiment. Okay, so we have broth in these flasks. Okay, and this is a special flask. They call it a gooseneck flask because the neck of the flask has this long kind of gooseneck curve, and it's open, right? So this is an opening from this end all the way through to the inside. Same idea down here. Okay, and so it has this broth, and time, lots and lots of time goes by, no microorganisms started to grow here. Okay, same idea down here. So he boiled it. Why do you think he boiled it first, I guess? Okay, so why boil? All right, so this is going to kill everything. So we know that we're starting with a broth that has nothing alive in it to start. All right, we're starting with broth that might have nutrients in it, might have like sugars and things, but it doesn't have anything alive in it. All right, so we boiled them both. Okay, this one, waited, no growth. This one, boiled it, killed everything inside, and then he broke the neck. Okay, so it has this opening to the world. And over time, it got all icky and cloudy and had that microbial growth in it. Okay, so what did he learn? Okay, so the argument against Reddy's experiment, okay, was that there was something essential about exposure to air. And they're like, well, this one doesn't have exposure to air, and maybe the gauze stopped the air from getting to it, and that's why there was no maggots. And so this kind of countered all those arguments. The broth was still exposed to air. Okay, but the microorganisms got caught in the neck. They kind of collected right here. Okay, in that kind of low spot. So they could get in through here with the air, but they didn't have legs. They can't crawl, so they couldn't go up and down and get into the broth. Okay, so they got caught in that curve of the flask's neck. Okay, and he was able to wait not just like a while, but like years and years and years. Room temperature, and this broth would never ever spoil. Okay, and so they kind of realized the value of this process, this boil it and seal it, and that is pasteurization. Okay, so if you ever look closely at a container of most kinds of cheeses or milk or yogurt and pretty much anything dairy, it's going to say pasteurized. And that just means at some point in its production, it was heated up really fast and then cooled back down again. And so it was heated up really fast in order to kill anything that was alive in there, any bacteria. And so then it can keep good a lot longer and be safer to eat. Okay, so food is heated quickly to kill bacteria before being sealed up. Okay, so for the most part, this is going to be dairy products that are pasteurized before they're sold. So again, why did he boil both flasks? This is to kill anything already in the broth. Right? Because we were trying to prove that life couldn't start from nowhere. So we had to start with a sample that did not have anything alive in it. So we had to kill anything that might be alive in it to start. Alright, real cells. Okay, so over millions of years. This is another one of those you kind of have to buy into the idea that Earth is really, really old. Okay? Wait a second. Trying to skip a page on you. Sorry about that, guys. Okay. So we have this idea, again, always supported by evidence, biogenesis. Okay, so this would be the alternative to spontaneous generation. See that gena in there? Like generation, genesis. So instead of life arising spontaneously out of nowhere, life is arising from bio, which means life. Okay, so this is the idea that life comes from life. Okay, but, big but here, this does not answer the question about where the first life came from. Okay, this idea makes sense once we have cells. Once we have living things, we see that cells come from cells, but where did the first cell come from? Okay, and the reality is we still don't truly know, which is fine. Okay, but we do have a few experiments that are definitely worth talking about. So we had Miller and Urey, that's their last names. This is the focus of that Edpuzzle from yesterday. They replicated early Earth with water, methane, and a few other gases believed to be present on Earth. Okay, so they had their fancy glassware, and they put the things in there that they only thought would have been, that they thought would have been present on early Earth. So there's, not a lot of oxygen and no living things in the flask. And they simulated lightning, which would have been present on early Earth. There's no reason to think there weren't storms if there was, lots of water and oceans and things. They found, after not that long, again, only a few days, okay, they found the precursors of sugars, amino acids, and other organic compounds had formed on their own. And these, they used to think, before this experiment, okay, were only found from living things. Okay, so I'm going to say usually associated with life. Okay, so before this experiment, they thought these kinds of molecules could only be made by living things. And they were able to create them in the lab under conditions that early Earth was probably like. All right, so how then do we go from organic compounds to cells? And this is where the evidence, we don't have it, right? We know that life has come from somewhere, but here we go. Okay, just as oil forms around droplets, if you ever drop a drop of oil on a pan of water, it forms a round droplet. So if we have the molecules of life, they're going to kind of naturally form spheres, and they call these things that are kind of spherical, but they aren't really alive. They don't have a metabolism. They are not able to do all these reactions of living things, protocells. Okay? And no, these would not be considered cells yet. Okay? But they have all what I like to call the ingredients in place to have cells kind of come from this. All right, now to the page that I was trying to skip to before. So now, real cells. Okay, so how do we get from a protocell, which is just kind of a sphere of molecules, to an actual cell? Okay, so it's this key idea. Over millions of years, we have lots and lots of time. These protocells may have developed into the first cell. And if you're like me, you're thinking, well, that's astronomical, that's crazy. But you remember, you have to think, this only needed to happen once. Okay, so in millions of years, in all of the oceans, we have to have things kind of come together correctly to make a cell one time. Okay, after that, biogenesis makes sense and can take over. Life comes from life, cells making cells. I think the first cells were probably similar to existing prokaryotes. Especially Archaebacteria, which remember was one class of bacteria. And I guess this is our reminder here. Prokaryotes are single-celled organisms, and remember their key is they do not have a nucleus. And remember, they don't have any of the other organelles really either, right? They don't have mitochondria, they don't have chloroplast, they don't have ER. They're just really, really simple. Right, they're a membrane, they have some genetic info, and ribosomes, and that's almost it, right? We have our two classes here. We have plain bacteria, which are the ones that are pretty much everywhere, and then we have the Archaebacteria, which are, again, this is kind of the ones they think are more similar to the ancient living bacteria, but they can live in crazy places, okay? Places that are super acidic, places that are super salty, places like hot springs and deep sea vents. Okay, these are places that no other living things can thrive in, and these Archaebacteria can live there. And so it's kind of nuts. Okay, so again, these are kind of conditions that are like early Earth. Okay, hopefully, okay. All right, so what about organelles? Okay, so how do we create those organelles that eukaryotes have? Okay, so again, we have another idea here. We have endosymbiotic theory. Okay, so endo means inside, right? And symbiosis is, remember, when two species live closely together. So we've got living together inside theory. Okay, so this says mitochondria and chloroplasts are actually descendants of prokaryotic cells. Okay, their ancestors were separate cells. Okay, and at some point they were enveloped or taken in by a larger cell that, rather than digesting them and using them for nutrients, didn't, right? And those bacteria, some of them would have been photosynthetic bacteria, and they became chloroplasts, and some of them were really efficient at using food and creating ATP, and they became mitochondria. And so, again, if you're like me, you're like, well, that seems a little far-fetched. What evidence do we have? So we have some evidence here, right? So this is later supported by some ideas here. Okay, so mitochondria and chloroplast have their own DNA. Okay, so inside a chloroplast is chloroplast DNA. Okay, inside a mitochondria is mitochondrial DNA, and it is separate from the DNA that's inside your nucleus. Okay, just like remember, we're thinking that they were like a prokaryotic cell. Well, if they took in a cell, that cell would have its own DNA. Okay, so not only do they have their own DNA, they have their own ribosomes inside. Again, just like a prokaryotic cell would have had DNA, a prokaryotic cell would have ribosomes inside. Okay, and this is the one that I think is maybe the most interesting. They divide on their own. Okay, so all the other organelles, when the cell divides, the cell has to kind of manufacture those organelles from scratch after the cell divides. But mitochondria and chloroplasts, they divide on their own, right? They copy their own DNA. They go through their own kind of division process, completely independent of the rest of the cell. Okay, so those are kind of our supporting ideas for this idea that organelles, especially those two, came from that they used to be prokaryotic cells. Okay, so endosymbiotic, remember endo is inside and symbiosis is kind of living closely together. So hopefully that term makes a little bit of sense. So all right, that's the end of chapter 14. Enjoy.